Alright, so our second last video for this chapter is actually on stem cell therapy as another alternative apart from gene therapy to more or less correct and hopefully in the future heal gene mutation or diseases associated with gene mutation. So we know that stem cells has many uses, as hopefully you still remember from chapter 5. Stem cells offer the opportunity for transplanting a life source for self-regeneration. Apart from that, we know that because stem cells basically has many uses, it allows specific tissue to regenerate throughout life. And we know that again, the specialty of stem cells is that it is able to self-renew and differentiate into any other types of cells or tissue regardless of where it is from originally. So these are some of the sources of stem cells, including sperm embryos, clone embryos, aborted fetuses, umbilical cords, adult tissue or organs, and cadavers. And by cadavers, meaning that any kind of cadavers or bangkai from animals. Now, stem cells basically are divided into two. We have embryonic and adult stem cells. For embryonic stem cells, usually cell lines are established from the embryos basically right after fertilization. So the embryos have to be separated into individual cells. And of course, again, regardless, endless amount could be obtained just from one single cell, okay, with controlled environment. If you know how to grow them in abundance and you have an established protocol. So most embryonic stem cell lines in use came from mouse embryos because it is generally and fairly easy to generate them in vitro. The second one on adult stem cells, whereby the cell lines are isolated from mature cell or mature tissue of an organism regardless of the animal. Cell lines may be used to form other tissue types. So embryonic stem cells most of the time could be used for adult stem cells, but the stem cells most of the time is used to form other tissue types. So we know that stem cell therapy is another form of alternative. So this goal of the therapy aims to repair damaged tissue that could not heal itself. And apart from that, to transplant stem cell into the damaged area, basically as the first method or the basic methods for this therapy. So transplanting stem cells usually requires some direction for growing new healthy tissue in place of the damaged ones. And there is a certain restriction to this technique as well, as you will see in a while. So how do stem cell therapy works? First and foremost, same with gene therapy, you have to make sure that you know the criteria that fits for this therapy. First and foremost, problem definition. See the similarities with gene therapy, you need to know the criteria first. If you can find out the cause of the disease, which cells are affected and how it can be treated, then it would be good enough. If not, find out more about it. Secondly, finding the right type of stem cell, because there are varieties of stem cells from different sources and whatnot. So you need to know which stem cells can differentiate into the appropriate cells, because even though stem cells could differentiate into any other cell types, but some of them may not be that efficient or may not be smooth running or may basically go through a problem. So you need to make sure of that. You also need to evaluate every types of stem cells to make sure that you don't encounter a problem later on. Third criteria, match the stem cells with transplant recipient. So immune system could reject stem cell transplant. So this is again the number one, I would say enemy to any therapy. So a close match between stem cell transplant and recipient should be checked. Cross match should be done. So this is done by tissue typing test with both individuals' blood samples. And bear in mind, even though they may be sisters or twins, sometimes it may not be compatible. And criteria number 4, deliver the stem cells to the right place. So of course this requires surgical procedure. Stem cells usually you couldn't use in vivo by IV drip, it's not possible, usually requires surgical. So image the projection and the procedure is carried out for precise delivery. This is to ensure that you minimize the risk of rejection and also any complication later on. And criteria number 5, ensuring the transplanted stem cells work. Of course, definitely. So again, as with any other therapy, not just gene therapy or stem cells therapy, no, there is no guarantee of actual function of transplanted stem cells. So the patient's progress usually are monitored day by day. So in terms of stem cells, there are two types, direct delivery and cell-based delivery, which means that, but both of them, okay, still have to be delivered via surgical or operation. So in direct delivery, as you can see over here, of course, anything or any modification of your therapeutic transgene again needs to be done. So you do it outside, okay, in vivo first. Then you choose the right vector, and most of the time this is well done using virus vector, and then okay injected into the patient, which is basically delivery. As compared to the cell-based delivery, whereby okay your therapeutic transgene okay with the vector incorporated into the vector is basically being grown in the lab first, right? And of course, okay, later on, okay, they are combined okay, with the therapeutic transgene from the cell, the patient itself. In the labs, okay, they are more or less mixed together, and well, they could be modified or multiplied or basically made into any other cell types, and then only re-administered back to the patient. So the hierarchy of stem cells are as such. So we know that okay, it could come from any types of sources. So well, most of the time there are totipotent cells. These are cells that are self-renewal, could renew themselves, and they are capable of dividing and developing to form a complete mature organism. There is also a pluripotent cells, which we learn in transgenic animal. It is also self-renewal, and it is capable of developing into many different cell types. So from there, usually okay, it is divided okay, into blood stem cells. These are usually specifically used to differentiate into red blood cells and white blood cells, and we know there are varieties of white blood cells. Apart from that, the pluripotent cells okay, could be developed to form muscle tissue or nerve or bone or other tissue that you desire, provided that you know the technology, you have the procedure to form it. So these are the 6 stages of stem cell therapy. I'm basically taking an example from a multiple sclerosis therapy from the website of basically UK NHS actually. So first and foremost, stem cells are moved from bone marrow to bloodstream using chemotherapy and synthetic growth factor. So this patient basically the bone marrow are taken first. So the machine collects blood and separates out the stem cells just to remove any of the plasma. So the stem cells are frozen and then ready to return to the body later. So more chemotherapy again used to fully or partially wipe out bone marrow and immune system. This is to make sure that they don't attack against stem cells when it is being administered to the patient later on. And stem cells return to the body using drip. And later on, the body, hopefully, if everything goes well, recovers over a period of 3 to 6 months or sometimes longer. So any modification needs to be done during this time. So while they are flushing, everything needs to be done, and when they are ready, okay, move on to step number 5. So how do we collect cord blood stem cells? So everyone has blood, right? And cord blood basically is the most useful. So what happens is that okay, cord blood are usually obtained from mother or when a baby has just been born. So the cord blood will be drawn okay from the clamped cord into a special collection bag by the doctor or the midwife, okay, be done, or the nurses. So what happens is that they make sure okay that it is going to be delivered as soon as possible. So usually it is done within 12 hours, 12 to 24 hours. So it is being delivered okay in dry ice, and basically once it has been delivered to the collection center or to the labs, so they will perform all necessary examination analysis of the blood. So this is just to make sure that the blood is okay, there is no disease, it is clean, there is no contamination, and so forth. So once completed, the red blood cells okay, that are not needed for preservation is separated. Those that need okay, for preservation will be stored okay, in another bag. Right? So and then again later on they are either being kept or they could be used for stem cell therapy for any kind of manipulation. If they are not used, okay, they are usually stored in liquid nitrogen storage tank at minus 190 degree inside secured facility. So as you can see, okay, cryogen tank okay, is being used okay, to store the stem cell. Now, for this kind of stem cells, it is basically used to treat leukemia or anything to do with blood cancer. Alright, so this is actually one of the example of how stem cell transplant research okay, has been done and postulated or basically has been tested for Parkinson's disease. So we know that Parkinson's disease is a disease whereby most of the time people misinterpret that it only affect the old people, because usually they see that old people, okay, when they are shaking, their hands are shaking, their bodies are shaking, whatnot. Okay, so that is actually an indication of Parkinson's disease. But young people could get it as well. If any of you knows, okay, an actor from the old times, okay, Michael J. Fox, he has it, okay, before he was 40. So it is not impossible for young people to get Parkinson's disease nowadays. So what happens when you have Parkinson's disease? Usually you have a dead nerve cells not producing the dopamine. Dopamine is being used, okay, to make sure that you have a smooth sailing, making sure that you could move about your, or you could do all your everyday function. You could walk probably, you could basically do everyday's work. When you have Parkinson's disease, because you have quite a bad shaking, so it's hard for you, okay, even to sip a drink, or sometimes okay, just to sit still. So what happens is that later on, okay, they do the modification in the lab, okay, using the stem cells, and then they implant the stem cells to produce new nerve cells that could produce dopamine, because obviously in Parkinson's disease, the basically nerve cells doesn't produce dopamine since it's dead. So later on, again, once they have ensured that the new stem cells, okay, could produce dopamine, so they would basically transplant it back into the patient, and hopefully, if it works well, the dopamine will transmit the nerve signals so that the patient could basically gain back, okay, the signal from the brain to making sure, for example, it could move about its finger without shaking too much and whatnot. So this is not a one-time cure. So usually it has to be administered for some time, and they have to monitor it closely because it's not suitable for all Parkinson's patients. Alright, so we know that stem cell therapy is another alternative for gene mutation, so it may provide promising future in curing diseases such as leukemia and Parkinson's. Again, these are still in its infancy. Up until now, it couldn't be sure, or it couldn't be said for certain that it could cure both diseases. So, of course, again, much is still needed to be done to improve the effectiveness of the stem cells, and of course, first and foremost, reduce the risk to the patients. And with the application to skin replacement, such as either for cosmetic or for any injury to patients, brain cell transplant, and also, for example, diabetes, a lot of work is actually currently ongoing to widen the potential of stem cell therapy in medical field. So you may wonder, why diabetes? If you know, diabetes is basically affected by the insulin production, and insulin is being produced by your, basically, a gland, again, near your thyroid, again, basically towards the base of your neck. So because it is produced by a thyroid, basically there is no pills, no medicine that could cure it, because you couldn't control the production or the secretion, okay, uh, from the secretion of the insulin, simply said. So using stem cells, okay, you basically could improve, okay, the production, because especially for people with diabetes type 1, whereby they don't produce insulin at all. So they could basically hopefully modify that so that insulin could be produced, and the person could basically have a normal life. They don't need to inject insulin every day to make sure that they could survive. Alright, so that's it that I have for stem cell therapy. If you have any question, just post it underneath this video, or we could basically just explore it and discuss in detail on Tuesday.